third live session, the third and final session for FGVC7. We're happy to see everybody here. And I want to first recognize some of our organizers who are here. Oshin Macaud, Sabransu, Grant Van Horn, Christine Kaiser-Chen, myself. We're grateful also to have many others who have helped us on the team organizing some of our competitions. And we have currently two of our, there's Barbara, perfect timing. Hi, Barbara. So I'm going to briefly introduce everyone. We have, and the talks you can watch offline. Those are on YouTube and also on our, linked off of our website and off of CVPR. But we have three talks that hopefully some of you are able to watch in advance, but we're very fortunate to have most of our speakers here with us. Olga Rosikovsky and Angelina Wang, and Barbara Ambrose is just joined us. Unfortunately, it looks like Andrew Farnsworth may not be able to join us, or will do so briefly. We're hoping he'll be able to, but may not be able to. So to start things off, I'm going to give each of the speakers just a minute or two to introduce, sort of for people who may be joining us and couldn't watch the talk in advance, give sort of a 2-minute summary or something of the talk, just so that people have the context for that. So Olga and Angelina, can I turn to you guys first?
Sure, sounds good. I'll give a broad overview, and then Angelina can talk about the particular tool. So that's how the talk is structured as well. So at a high level, so I'm an I guess I'll introduce myself. So I'm an assistant professor at Princeton. So my background is in building visual recognition systems, but also collecting data sets. So doing a lot of crowdsourcing work, particularly during my PhD. And so one of the research directions right now in my lab that we're looking at is the question of fairness, questions of fairness within both the recognition systems as well as data sets. And so there's a number of papers that have come out, including one at CVPR this year, some looking at the models and sort of doing bias mitigation of models, and some looking at data. And one of the things that we've been finding in the on the data side is that it's very difficult to sort of do bias mitigation in a large scale data set. And I think a lot of the answers that have sort of come out of the fairness, accountability, and transparency community, which is this growing community with a new conference, the Fairness, Accountability, and Transparency Conference that was started a few years ago. The answers that have come out of there have been of the form, the creator should be looking at their data and examining their data and making sure that the data doesn't have problems. The issue is this applies when your data is 200 images or 300 documents, and it doesn't really apply when your data is ImageNet or COCO or any of the actual large-scale data sets that we're working with in computer vision. And so this is where Angelina's work comes in, which was the main part of the talk, is in building a tool that's actually able to reveal some biases in a data set sort of semi-automatically and present it to a user to sort of make decisions about and potentially make modifications to the data set. So I'll pass it on to Angelina.
Hi, I'm Angelina. I'm a PhD student working with Olga. And the tool that we built right now is kind of looking at biases along 3 dimensions. So one of them is object-based. So for example, maybe certain objects only appear in like the same context every time, and it's possible that models trained on this data set won't be able to recognize the object out of context. Another axis is gender-based. So for example, different genders might be represented very differently in a data set, and it's important to realize when the biases present could propagate into the models and like amplify them in ways that mirror biases in society. And then the third one we look at is geography-based. So this includes things like how a lot of data sets primarily come from like images collected from Western countries and have trouble recognizing objects from other kinds of areas that are underrepresented.
Great. Thank you, Olga and Angelina. We're excited to have you with us. Barbara Ambrose works for the New York Botanical Garden, and I'd love to give you a couple minutes, Barbara, to introduce everyone to what you presented in your talk, just briefly.
Yeah, hi, good afternoon. Barbara Ambrose. I'm a scientist at the New York Botanical Garden, and I study patterns of plant diversity at all levels, from the molecular to global patterns of plant diversity, which relates directly to the data set that was presented in the Herbarium 2020 Challenge, which was more than 1 million herbarium specimens representing more than 32,000 species. And our interest in developing machine learning algorithms to identify these specimens automatically is that we have in our herbarium alone 7.8 million specimens, and this includes plant and fungi. And we have 400,000 known plant species. And there's an estimated 80,000 plant species still to be discovered. And describing those species is a very cumbersome task. If you're a taxonomist and you're describing a new species, you really have to have this encyclopedic memory about what you've seen to determine if something is new. And there's this idea out there that of those 80,000 undescribed plant species, 50% of these are probably already present in herbarium collections. And there's few, very well-trained taxonomists who can curate a collection. So in the first instance, we'd like to use these models to better curate specimens in herbaria worldwide. There's 3,000 herbaria worldwide. And we also like to further develop the models to help to identify unknown specimens, which is not the way the models are developed. So we're really interested in sort of anomaly detection. How does it know when it doesn't really know something? Like, how can we identify when a species is actually unknown, undescribed species?
Perfect. Thank you, Barbara.
Sure.
We have an interesting, and Andrew unfortunately wasn't able to be with us. I don't think he showed up. Andrew works for the Cornell Lab of Ornithology, and the Cornell Lab does a lot of work on both visual and acoustic recognition of birds. A lot of fascinating science that they do. We're sad that we couldn't have him with us, but we do encourage you to watch his talk. And so we have Olga and Angelina, who talked about sort of various ways that we see bias in our data sets, and Barbara and Andrew both talked about sort of these natural world applications, identifying and discovering new species of plants and identifying birds, especially sort of acoustically. And so I wanted to start with a question, I guess for Barbara. I recall, I think you may have mentioned this in your talk as well, but from prior time talking with you, that it can take almost 30 to 35 years to identify from the time a plant may be discovered or collected till the time that it's actually identified. Is that the typical lifespan? Is that kind of average, or can you tell us a little bit more about that?
So that's been reported in the literature as a sort of average. So at the New York Botanical Garden, we are still going out and collecting specimens. And if you don't have an expert or if you're not the expert in the family that's done the collecting, what happens is a lot of those specimens go first into, they're pressed in the field. They're then taken, or shipped to the garden. And we have a room right now, there's a cold room of specimens, they're pressed in newspaper, and we have 30,000 specimens sitting in that room. So they have to get mounted, and label data is put on there, like the collector, and they may have some idea about what the family is, but if those aren't sent to an expert, they can sit around, and so it can take 35 years from field collection to species identification.
So in that process, all the steps leading up to, sort of mounting and so forth still have to happen even if we have a computer vision system, is that, or can you just recognize it out in the field and recognize that's something that we haven't seen before? Do you have any?
If you're an expert in the field, right? So if you know the species that are out there, this like my colleague, who studies the family Melastomataceae, he's very aware of his family. It has, maybe 5,500 known species. And so when he collects, he might have an idea that something is new, that it might be morphologically distinct. And so he might tag that one. And even though we have this expert, Fabian Michelangeli, as an expert at the garden, we still have 6,500 melastome specimens without an identification. It's tricky business, yeah.
So I have a question. I don't know if the panelists are allowed to ask questions or not, Ryan. Is it okay?
Absolutely, Pietro.
So a follow-up question for Barbara. Then I also want to hear from Olga and the others because it's very interesting. But so let's imagine a future where computer vision people do their job, and so you can go out and with your iPhone, you go out in the field, you take a few pictures, and our computer programs recognize the plant if it's already described, or they tell you, look, this has not been described before. It might even compute a 3D model of the plant immediately so that you can view it however you want. It's like having it in the herbarium. It's wonderful. So let's suppose that problem is solved for a moment. Okay. Now, can you walk us through in what way does the world change as a result of that? How is the world becoming a better place? What is it that you guys are going to do that is going to make a difference?
Yeah, sure. I'm happy to take that question, because this really goes towards, the crux of the work that we do at the New York Botanical Garden. It is about discovery and exploration. And there's some 80,000 unidentified plant specimens out there. That's the basis of all of our food, all of our shelter, a lot of medicines are based on plant products, and we know we're losing these habitats quickly. So maybe of those, if 50% of unknown specimens are in the herbaria, another 50% are somewhere in the world. So if we could, in this is my sort of dream world, be able to link with the specimen data that you get, right? When a plant is collected, it has coordinates. It also has the time of the year, and plants are usually collected when they're reproductive, because they'll have many more characters to help to identify them. So then when a plant is flowering and fruiting, which is where all of our food sources come from. And in the herbarium, we have collections that date back hundreds of years. So my ideal end point would be to layer all of the different data that we have. So specimen data, where are the plants? Where would we expect to find them? Soil data, climate data, and start doing predictive work in trying to estimate, okay, the same plant could be collected multiple times over decades. When it's flowering and fruiting, does it change? Does this tell us something about how habitats are changing, where we should be planting crops? And also, if we have an idea about where species diversity should be, that would also help us to target areas of exploration. Where should we be focusing our efforts to go out into the field, which is no small feat. I mean, going out and collecting plants is a lot of hard, long work. So ideally, what it would help us to do is that was the long answer, short answer, be able to focus our exploration efforts, be able to determine how habitats are changing over decades to help us better understand, which plants are surviving in certain climates, which plants can grow in certain places, which plants are new that could be potential crop species, which species do better in the boreal forest, because those are the first ones we're losing to climate change as well. So there's a lot of different, I think, use for these models. Right.
Okay. So Ryan, may I ask a follow-up question, or so you moderate me? This is very interesting for me, but I don't want to monopolize it too much. So let me ask you the follow-up question. So the way I understand it then is there is a physical world out there, and there is a giant model we're building where in the limit we know about each specimen of each species. Where is it in this moment, and which cycle of its life is it at this moment, right? So I'm just making a little bit of an extreme vision. And computer vision is helping connect the two. So we have the model. It's like, let's take weather forecasting as a parallel. So we have a model of climate, we have a model of the weather, and we have sensors like balloons and thermometers that connect the virtual cyber model and the world. And so we would like to get to the same point with plants. Is that more or less what we're talking about? Okay. Okay, that's great. It's very clear.
Great. Thanks, Pietro. Question for Olga and Angelina. So curious in the work you've done, so with understanding and mitigating bias, when we collect data sets for fine-grained recognition in particular, these tend to be long-tail distributions. So in some sense, they're inherently biased. One of the things that we observe, for example, in a data set like iNaturalist, where citizen scientists are collecting data just in their day-to-day lives in some cases, others are out in the wild and so forth. But we tend to have a lot of data, for example, in the US and relatively little in some of the sort of third world countries and other parts of the world. Do you have any thoughts on, I mean, particularly as it applies to this type of data, how we might be able to help mitigate that, or maybe have models that are more robust to those?
I mean, I'm happy to jump in, and then then we'll pass it off to Angelina. So I think there is this sort of not necessarily like misunderstanding, but there's sometimes a sense with like fairness that the answer has to be that we have to do, you know, everything perfectly fair, that like our models need to be perfectly fair with respect to, you know, and then you sort of provide some kind of mathematical definition of fairness. But I think the reality is it is a process. It is sort of one of those things that's like nothing is ever perfectly fair, because what does it even mean to be perfectly fair? But a lot of this is about like being aware of these patterns. So I think the biggest issues really come up when you're not aware of the patterns. And like I say here, okay, so you know that the data is long tail in a particular way. And this is actually gives you, you know, a great leg up over many other data sets where many other types of biases where we actually aren't aware. We have no idea what's actually hiding in there. But once you know that there's this long tail, then comes the question of, you know, do you bring in computer vision models and do you solve it from a modeling perspective? And one of the, I think, really cool things about fairness work right now is a lot of it actually reduces down to a reformulation of the problems that the recognition community or the machine learning community has been working on for a while. It's just sort of a slightly different look on that. Or if you know that there's a long tail in terms of geography, right, you can think about, you know, is it something that you as data set creators are doing that are injecting that bias into the data, or and like is there some steps that as data set creators you could take to be more more inclusive, or sort of think more broadly about where this data should come from, or how to reach out to people in other countries, or sort of why aren't they contributing to this? Are they not aware? Do they you know, have no interest in contributing to this? Do they not? I mean, what is the what are the reasons? And then once you're aware of it, you can come up with solutions to begin addressing that. Jillian, I'm sure you have lots of things to add as well.
Yeah, I agree with Olga that I think like thinking about why the data set collected was long-tailed and addressing those issues could be very helpful in encouraging like different people from different areas to contribute more data. And then also kind of like thinking out what kinds of distributions that are long tail are problematic depends a lot on the application of what the models trained on the data set will be. So like some areas of the data set that are long tail that might not be as big of a problem as others. So specifically addressing those that are like relevant to the application would be the most important to do.
I had a follow-on question kind of related to this, probably for Angelina, but maybe Olga as well. So in your talk, if people haven't seen it, you presented this really cool kind of diagnostic tool called Vibe that like pulls out some of these biases that are in data sets and kind of allows you to peer into it and get a better understanding. And so if I understood correctly, these things are all predefined, or these relationships are predefined, or the types of biases that you're looking for are predefined. And I'm wondering, like, do you have any comments or thoughts on the idea of how we might discover those, or how this might be an iterative process, for example?
Yeah, I think that like it seems like with every application of computer vision or machine learning to the world, like people find new biases that are problematic that people hadn't thought of before. So it seems like each time this arises, you could kind of like work in how to discover these kinds of biases back into the tool and have like a big feedback loop and cycle. Because it's like, it seems impossible to be able to predict all the different kinds of biases that could arise. It seems like something that kind of will come up.
I guess I was kind of thinking about it again, maybe not to always map it back to these naturalist settings, but you know, let's say we only have pictures of pigeons in cities or cows in fields and never on the beach, and you know, it might not be immediately obvious to us what these biases are, these correlations, but I'm not sure how we would go about, yeah, surfacing them or discovering them, other than seeing when our models fail, if that makes sense.
So one example of a really strong bias that consistently gets learned in my camera trap photos is daytime versus nighttime. So there's some animals that are really nocturnal, and then if you see them during the day, the model like consistently will just like really struggle to learn it, even if it's a common species, because it's acting sort of non-nocturnally.
Yeah, so that makes a lot of sense. I mean, to add on to what Angelina said, one of the things we do is we do run pre-trained models on the day. So for example, something is pre-trained on places, so that won't get at what Sarah's mentioning about day versus nighttime yet, but we would get at the scene category. So we would actually reveal that, you know, these things tend to occur more often in particular types of scenes, and that would be something that would get surfaced by the tool. And so certainly, you know, there's definitely opportunities to bring in, you know, either whatever existing annotations you have on the data, or if not, whatever existing models there are. And the models, of course, are going to come with their own biases. And of course, it's all going to become a chicken and egg problem. But at least to sort of start getting at surfacing what we can out of the data.
Yeah, absolutely. And yeah, the data I work with, because the cameras are static, actually the bias is like just almost insurmountable. It just is so hard because the sample efficiency is so low within a static camera. And so that's like a huge portion of my work, is just trying to figure out how to overcome that and work on new static cameras.
So one thing I forgot to mention, anyone who is participating, joining us, we do have the chat window in Zoom. If you want to ask questions, you're welcome to do so there. We'll try to address those as well. One other question just occurred to me, kind of connecting the work that Olga and Angelina have done with what Barbara mentioned, that one of the things that they're working on is discovering new types of plants, and that that's sort of an open set problem, right? Inherently, it's an anomaly detection thing and so forth. I wonder if there's anything, Olga and Angelina, that you guys might be able to add about open set type problems. Sort of how would you recommend addressing that, particularly in a domain where things look very similar?
Yeah, so I'm thinking of a good answer. That's yeah, I mean, I think that's definitely difficult. Whenever I think of that, I would think of when I think of that in the fairness context, I start thinking of long tail immediately. So I know OpenSet is different from that, but I start thinking about, you know, the sort of, when you find something or reveal something in a data set that doesn't look like the other, more often than not, what that means is that it's a long tail distribution, or sort of the subclasses within a particular class are distributed in a long tail way. It's sort of harder to, yeah, I think it's, yeah, I think in the kinds of data set that we have worked in, more often than not, it's basically reduces to a long tail problem, but that's, yeah, that's different.
So in the case of a long tail problem, would you advocate, I mean, part of what you've worked on is sort of mitigating these biases. Would you advocate sort of changing the way that we collect data to kind of make that problem easier, making our models more sort of robust or resilient against those, in those types of settings? What types of steps would you suggest that we as a community take in those kind of long tail cases?
Yeah, so 100%. I would actually, I think Angelina was spot on with the question of what is the application. And I think actually, you know, one of the things that the fine-grained community honestly has been so, you know, several steps ahead of kind of the rest of visual recognition is that everything is related ultimately to an application. It is driven by and related to an application. And I think the sort of basic level recognition community is actually moving very much towards that now. It seems to be like it's less and less about, you know, oh, let's just, you know, build models that work on Pascal and then figure out what we can do from there. It's more and more sort of building particular applications and sort of focusing on particular domains and on the data coming from there. And this is why actually the fairness issues are starting to become more and more relevant, because it's starting to really make its way into final applications. And so I think the question would be, you know, exactly as Angelina said, is what is the application? And if the application is, I would like to recognize birds in daytime, do you care that a few of your data, you know, some of your data set is nighttime and that's the long tail? I mean, do you care? Maybe you don't. Like, maybe that's not a problem. If the birds you're seeing are primarily daytime, that's fine. Similarly with geography, right? If your application is going to be deployed in the US, you might want to, you know, say something when you're deploying it about how this is not going to work in other countries. But other than that, you know, seems fine. If the application actually, you know, if the plan is for it to be deployed in domains other than what the training set represents, that's when you have a problem. And then, you know, I think the solution will always be a combination of data and models, depending on which one is easier, honestly. Like it's always kind of a chicken and egg problem. And in some cases, the right answer is, well, let's just collect more data. And it's, you know, simple enough. And especially if it's in a setting that's not a fine-grained setting where it's just, you know, you throw it on Amazon Mechanical Turk, you don't need experts, it's fine. You can go grab a lot more data and get it labeled. In a setting where, you know, data may be scarce and it's just maybe near impossible or very expensive to collect it, then you turn to the algorithms. No, Angelina, if you wanted to jump in with something.
Oh yeah, I agree with all the things you said.
Okay, great. Pietro, do you have a question for them?
Yeah, I do. So for Olga and Angelina, so it's both a comment and a question. So while I was watching your talk, I amused myself at thinking, trying to guess, you know, what would be between male and female, very sure ways of getting all males or all female photographs out of a search in Google. And so, if, for example, garbage truck driver, mostly male, you know, surprisingly so. Beautician, it's also exclusively female, both the practitioner and the customers. It was almost ridiculous. I thought that there would be some men who do beautician, but that none is represented there, and so on. So on the one side, I think it's great to discover these patterns, also not only for computer vision, but for sociology and for understanding how society works and all of that. However, as a long-term goal for our field, I think that if we view having unbiased data sets as a means to achieving computer vision systems that work well, we're shooting too low. I feel that if I've never seen a male beautician, but if I saw one, I would have no problem recognizing one, or a female garbage truck driver and so on. Or if I saw a sumo wrestler who is pole vaulting, you know, never seen one, but of course, if I saw one, I would recognize it, or a penguin in my bathroom and so on. So I think that we have to aim high and build computer vision systems that can learn from biased data sets and can generalize and can recognize any pattern, you know, with or without bias, but they're not. And so that's the AI goal. We shouldn't think that the world ends with deep networks that require 1,000 training examples, and if you don't have them, you're stuck. However, so one last comment, but I'm curious to hear your one place where I think analyzing for bias is really important is the test sets, because those are the ones that tell us if our algorithms are working. And right now, I find most of the measurements I see, both in the FAT community and in the computer vision communities, very disappointing. There are huge correlations in the test sets, and people make claims about bias that are not well supported. It's by these data sets. So that's a place which I find particularly troublesome. And so I'm curious to hear whether you see things the same way, or if you have other ideas.
Yeah, I think that's something a lot of people are doing now when they report test results, is disaggregating the results by like different metrics. Like maybe they'll have one accuracy for like females and one for males, to get at looking at the difference rather than like the total accuracy. And then also for the example you gave about the beautician, I think that it's a lot easier for us as people right now to learn these like the ability to generalize to even saying a male beautician and like knowing what their job is, because I guess like when we as humans learn, we have like a lot more feedback that we're learning from. But if you give a computer vision model like just a photo and every time it sees nail polish and a woman you label as beautician, it's not really clear, like you're not telling it what parts of this image make it a beautician. So it has to rely on these correlations, and like we as people I guess are smart enough to figure out that like what it's actually referring to.
Right. So just to explain better what I meant with test sets. So if you divide up results, error rates for male and female, let's suppose that you had visual recognition, which is a topic of great interest right now, and you found that males are less well recognized than females, right? So the temptation is to say, oh, the algorithm doesn't work so well for males. We need to collect more training examples. But maybe the male population, there are many people wearing a beard, for example, and then they're difficult to recognize because of that. And if you take people without a beard, so females without a beard, which most of them will, and males without a beard, then maybe the performance is the same. And so there are all of these funky correlations in the data sets. And if we're not careful, they pollute our measurements, and we don't know what we're talking about when we give out numbers.
Yeah, that's a good point. I think that a lot of times, like what we think the bias is actually a proxy for something else that is the real bias. And a lot of times it's very hard to figure out like what the true biased correlating factor is.
Right. And so there I find that your work is going to be particularly useful.
Great. Thanks, Pietro and Anjana, for your thoughts on that. We have two questions from our audience. Jongchi, do you want to ask your question?
Yeah, so I saw your paper. Maybe this is unrelated, but I saw your paper on toward fairness, and seems like the best one is to have a domain-specific model. But what if a domain is hard to distinguish, it's more continuous? And how do you know what the test time, what domain it is?
Yeah, so that's a great question. So this is a question presumably about the CVPR paper that's different than what we're talking about here. So the short answer is, in that paper, we're assuming we know the domain. And this is, there we know that this is, we model this as color grayscale. We also do, domains that are male versus female, pretending that gender is binary and using those labels. We do experiments with domains that are CFAR versus ImageNet, so sort of depending on where the data distribution comes from. So we make the assumption that you know the domain. I think the question about what happens when the domain is continuous, or what happens when the domain is not known. So to the question of what happens when the domain is not known, when we actually started this paper, our plan was to spend a couple of months in the setting where we know all of the domain labels, and then move to the really interesting case where we don't know that. And what ended up happening is that it turns out that the problem is really hard, and like much harder than we expected, even when we know the domain labels. And so we sort of stayed in that area, but 100% originally the plan was, okay, how do we automatically discover the domain, and what happens when you don't know the domain, and so forth. Great. So the short answer is I don't have a great question for what happens, how do you discover domain? That's future work, and what happens when the domain is continuous, also future work. So I think that's those are the two most most clear next steps out of that paper.
Yeah, thank you.
Oh, for sure.
We had another question. Deepti, would you like to ask yours?
Yeah, hey, Olga and Angelina. I really enjoyed your presentation and learning about Vibe. From your analysis, I'd love to hear your thoughts on how much of the geographical bias, specifically that you talked about, could be handled by different data sampling strategies that you suggest in your work versus and like that's the first question. And the second part is what kind of unique modeling challenges would the geo bias specifically open up?
I think that for geography, the biases could come in two forms where the same object might look very different in different countries, but then also the distribution of objects and how they co-occur might also be very different across different countries. And those would like both of these biases just kind of manifest in different ways in the models. So yeah, it seems like it's hard to come up with other ways to deal with this besides just kind of collecting more data from these underrepresented countries, because it's hard to extrapolate if you just don't have very much information in the first place.
Great, thanks Deepti for the question, and Angelina for your answer. Christine, you have a question for Barbara.
Oh, that's right. Hi. So my question for Barbara, actually also maybe Sarah as well, is I'm curious from your perspective, say Barbara, like if you want to start using kind of deploying these machine learning systems in production for you to say do like species discovery or species tagging, what will be an ideal workflow in your mind? Like, because sometimes we think that right now it seems to be a more or less manual process. Like there's a human expert that looks through almost all the record, but this doesn't scale very well for millions of records you have. But on the other hand, we might have, let's just use machine learning on everything and then somehow just assume it's true. But like, what do you think from kind of your particular domain? Like what's kind of the right way if you have an automated system that may or may not have some errors, and yeah.
Yeah, hi Christine. So one of the things that these models can be used for right away is the 3,000 herbaria that I mentioned. There's not a lot of taxonomic expertise. Some of these herbaria are very small. So if we could make these models, we need to optimize them to more species, but just to take them and to make it so that people who are working at herbaria can simply take their specimens and take a picture, run it through the model very easily and say, okay, is this specimen been correctly identified? Because once a species is identified as a plant species, I don't know if you've seen the labels, but somebody will come in there and change the determination of the species. So we are constantly curating and updating as we get more information about species out there. So the models right now are would actually speed up that process and actually make it maybe possible where it wasn't possible before to just curate existing data sets. For the data set that we presented, we have 7.8 million specimens, but maybe 50% are digitized. We had 2 million we thought we could put in the data set, but there is, we really wanted to make sure we had a very well curated data set, that when we said a species was a species, we were absolutely sure of that. And so we go through something, there's, different things that you can go against. We went against the plant list to say, okay, these are these things, but there's also a lot of synonymy in species identification, meaning there's lots of names and it's the same thing, but they're recognized. But to do that type of curation, we didn't have enough time for this data set, but I would think that would be something that would be amazing to automate, because it's just tedious. The data is out there, but you have to find where there's duplications and make a list that's very clear. This is the species we're talking about, and it includes these specimens. That type of information that would speed it up amazingly. And if we had more botanists, it would be much faster too.
I'm actually kind of in the same boat in terms of like what we can actually do in science right now with machine learning. I've worked a lot with these models, and just because the natural world is so long-tailed and because so much of our data has these biases that we're not able to disentangle well, I just don't trust it. So actually one of the things that I think we really need is basically like good research into how to best do quality control on the results of data on things that are unlabeled with experts where we're really optimizing their time. So like, what are the things that we should be triggering? And maybe that's, you know, catching some of these biases and like explicitly finding cases where this might be a problem. Another thing we've looked at is just like trying to use statistical measures of like how bad the image is, just like, is this have really, really low contrast or is it super blurry? In that case, if it's really high confidence, probably there's something wrong. But yeah, just like trying, even if it's just hacky tricks, like what is the best way to actually use humans sparingly but effectively to try to do quality control for our models. And this is something that I'm like actively worried about because I'm working with Wildlife Insights, and which is this, they're trying to basically build this large scale global data management platform for camera trap data. And one of the things that absolutely horrifies me is this thought that someone is not going to really understand that the machine learning is like not really right yet, upload their data, get species labels, take them blindly, you know, not verify them at all, and then start making science policy suggestions or conservation suggestions based on something that we know is not correct. So yeah, so I think that as it currently stands, I think what we really need to be focusing on is quality control. And like, how do you use humans effectively?
And it's not just, it's not correct, but it's going to be not correct in like biased in particular ways. So it's not going to make random predictions on the subset where it's wrong. It's going to make consistent predictions.
Totally, and those consistencies can be like specifically really incorrect. There's actually been some really interesting work in the ecology community recently that's looking at the effect of human bias or human sort of types of errors in identifications and how that's messing up, you know, the end results of some of these like population models or species distribution models. And so it's like, if we know that humans, which we're considering to be are like end all be all experts, if their errors are going to cause these problems, like of course our machine learning models are going to propagate those errors through as well.
So, but Sarah and Barbara, is it fair to say that unless we know the whole chain, it's a bit going back also to Olga's argument, unless we understand well the whole chain that goes from the observation in the field to a final decision that is made based on those data, so how is that going to be used? And we don't know it very well. We don't have a good idea of where the data is going to go. Then we really don't know what is the error rate that we can live with. And we don't know if we should aim for lots of species to cover the whole world, or we should aim for just a few, I forgot how they're called, but you know, signaling or, you know, charismatic species that we based everything on because they represent the rest, and so on. And so it feels to me, but maybe you know better, that we haven't thought through the whole chain between the camera that takes a picture and finally the law that is being passed to change some regulation or something.
I totally agree. And it's actually really tricky because what's happening along that chain is like 5 different areas of expertise. So you have like the expert in designing the field study, and then you have like the computer vision expert that's trying to get, you know, labels automatically. And then you have the modeling expert that's going to take those labels and turn them into some sort of model. And then you have the policy expert that's trying to understand how to take that and turn it into some sort of like conservation guidelines. And so for any one person to understand that entire pipeline, they have to master like 4 completely like different subject areas. I've been actually just really recently diving into the ecology modeling like ecological modeling literature, trying to get a better understanding of like if my model systematically produces more false positives for this animal, what would that mean, like mathematically down the line? Like trying to just probe it a little bit so that I can at least just get a better understanding of like which types of errors are more of an issue. But it's super hard. It's a whole like very, very long standing field that it's pretty hard to just dive into. And so I really think what we need is much better cross-field communication, like more collaboration with these domain experts instead of just trying to like understand really everything ourselves, like try and just share information better and learn how to speak each other's languages.
Yeah, and learn how to communicate confidence intervals down the food chain, right? So that everybody knows what confidence intervals they have.
This is another computer vision thing that we can work on, is like, how do we output probabilities instead of like random numbers that don't really mean anything? Because the ecologists would love to be able to treat the output of the model with like a confidence. Then they could actually just model that like within their, you know, species distribution model, for example. But it's like, people constantly want to use those as probabilities, and I'm like, oh, nope, they're not. Like we can try and do some like calibration, but even then, like maybe still not. So yeah, I think that's a problem.
So back to Pietro's point about like the need to understand the whole pipeline, right? So this is where, if any of you haven't seen the work from Tamid Gebru and from Margaret Mitchell, right? The data sheets for data sets, models for model card for model reporting. I had to look up the exact title of that paper, but so that's work around basically saying, you know, with every data set, we should be releasing a data card describing what is in the data set, what is the intended usage, what are the kinds of biases that are present in the data set, sort of where is this data coming from, what can you expect from it, and similar for models. And so that's kind of the first set of attempts to try to mitigate this problem, because yes, of course you can't always know what the exact pipeline will look like, but at least you can provide more transparency into the different steps of the process. So if you haven't seen that, definitely encourage you guys to take a look.
I had a question actually for Barbara on the topic of bias, but a different type, maybe like a domain bias, in the sense that if I understand correctly, these herbarium sheets you basically have these flat pressed versions of these plants. And yet when you get into the real world, obviously plants don't look like that. And so immediately this is a problem for our computer vision models, because I think we can't train on those and generalize to the real world. I'm wondering to what extent, you know, people that work in the botanical gardens, for example, can learn to identify species from those sheets and apply that knowledge in the real world, or for them also, is this a problem doing that mapping?
Yeah, so no, a lot of determinations for new species are made from herbarium specimens as flattened specimens. Yeah, so they're used. I mean, you know, they're obviously collected, you know, fresh. It may not be the person who determines the species from the herbarium specimen may not be the person who collected it, so they wouldn't have seen a live specimen of it. And we're also talking about, you know, we have specimens that are hundreds of years old that obviously we have no photographs for, and the people who collected them aren't around. So determinations are made, you know, it's kind of funny when you look at those herbarium specimens, you know, they're flattened, there's very little color on them. And it is quite amazing that you can determine species from those specimens.
Can you go from those, could you look at that sheet and then go out into the real world and be like, okay, yeah, that's the same thing?
If you were an expert in that family, yes.
Thanks.
Yeah.
So that's another, so it sounds like from the point of view of computer vision proper, you pose to us maybe two key problems. One is to be able to come up with a species recognition model from just one specimen. And so it's a one-shot learning of new species because you have only one specimen per plant. The second one is to do this mental translation between the herbarium specimen and the specimen out in the world. And I think both problems are very exciting for computer vision people. They're very interesting.
Yeah, I mean, nowadays a lot of the curators at the garden, they have hundreds of images that they've taken of plants in the wild. I mean, so from like the last, say, 20 years. And so they have all of these live images that also then, and they also have the pressed specimen. So it would be interesting to be able to go between the flat and dried specimen and the live specimen.
Is that important? Namely, with the digital cameras, you say there are now hundreds of photographs per species. Suppose that we burnt all the herbaria. I realize I'm saying something that makes you cringe, but would we have lost a lot, or within 10 years we have all that we want back in terms of digital pictures, and we don't have to worry about translating between the herbarium and the actual 3D shape?
Yes, so say you're going to burn down an herbarium is like saying you're going to burn down the Met and the Louvre altogether. You know, those are, you know, they're collections, they're historical data, and people do go back to them to learn about the history of their organism. I mean, it does go to, you know, now I think that, you know, a single botanist can really understand quite well their small family of plant species, you know, from hundreds to thousands. But how do you begin to understand that on a global scale? And when we're losing habitat so quickly, going back and forth from what you see in the field to what you found, what you can find in herbaria worldwide, when you start to look at those specimens, when they were, again, it's like the timing of collection and the location of collection, you still learn a lot about what that specimen looks like. And we can also, you know, I actually work in genomics, and a large thing that's really quite big right now and has been for quite some time is you can actually take that dried bit of plant specimen and extract genomic DNA and get a whole bunch more information out of it.
Great. We have a couple questions in the chat window that we should catch up with. Thank you, Barbara. Zhongqi, you had another question?
Yeah, my question is how to keep the model performance and also make it unbiased. Is it maybe have a domain-specific model, but then it go back to my first question of how do you detect the domain?
I think one thing to think about is that performance itself is like a very subjective measure. Like there's not like an objective, this performance is the best. Because even accuracy is like very dependent on the makeup of the test set, right? And like, so if your test set is all built off of one demographic and your model does great on that, but it does horribly on another demographic, which is what it will actually be deployed on, then this performance measure doesn't really mean anything. So I think that like performance and like fairness and bias, they all kind of go hand in hand, or are like very dependent on the final application of the model.
Yeah, I'd join in there. So I think specifically also like there's a difference between maybe the types of models that are predicting like some of this like sort of gender specific attributes, as opposed to like the models I work with where like you're trying to predict species identities. Like in that case, like there's not sort of any sort of, well, who knows? I don't think there's any problematic sort of like confounding factors in terms of like, you know, what species is this, and is getting the species wrong gonna like what problem is that gonna solve? I think it's more just like, if you know, and again, it depends on what you actually want the utility to be. If you know that you are really specifically trying to use this study to get like a very, you know, across the board accurate species distribution model across all the species that are seen, then honestly, like it might be better to let your model be biased and, you know, properly count the more common species better. And if you're gonna like sacrifice performance, you know, across like all the mule deer in order to properly categorize like a single example of another species, like in some cases that might be bad. In other cases, that's exactly what you want because you really care about that endangered species, and you actually just don't care at all if you have to look through like 300 more mule deer photos in order to get that single very rare species correct. So it ends up being this weird thing where like you really just need to come up with ways to evaluate the models based on your actual desired outcome. Yeah.
Yeah, I agree completely with what Sarah said. And it depends on, like, I think what the domain experts in that area say. So like, if you're doing something with humans, it might take someone that studies sociology to tell you which kind of biases like have this long history and are going to be more problematic than others.
So I'll jump in super quickly just to clarify, because I think this is a little bit of a shout back to the answer I gave on the previous time you asked about the domain independent paper. So I wanted to clarify a couple of things. So one is, at test time, you don't need to know the domain in that paper. So you integrate over essentially the set of domains. The second thing is that, so back to what Angelina just alluded to, right? So at training time, discovering domains automatically in some ways may not be helpful, because what you're trying to do is actually address particular types of inequalities in the data. And generally, when we talk about this from a fairness perspective, these correspond to disadvantaged groups. So this corresponds in some way to historical biases, historical issues. And so sort of identify them automatically may not actually be what you want. You want it to somehow correspond to some issues you know about and are trying to fix. And this is part of where then Angelina's tool might actually come in to show you the kind of biases that might exist in the data set, and then kind of have a human verify whether these is the kind of domains that you would want to correct for. And the last thing related, since we keep coming back to the paper, I did want to give a shout out to Zhe Wang, who's also here, who's actually the lead author of that work. So he's really the person to reach out with if you have more questions about that.
Yeah, feel free to like ask me later. Yeah, definitely. Thanks.
You're muted, Ryan.
Oh, sorry. And then feel free to reach me later. Yeah, thanks for, yeah.
Okay, sorry about, hopefully you can hear me now. Two more questions from the chat window. Aditya, you had one for Olga and Angelina. Maybe we'll come back to that one. Pietro, you had one for Barbara?
Yeah, it's not a big question, but so Barbara, I was trying to go back to what you were saying earlier. So you said that there are 30,000 species in herbaria. Am I correct? And you're muted now. So I'll complete my question. So my guess is that you're asking us to deal with about 100,000 species of plants eventually, right? So if there are 30,000 in herbaria, times 3, there may be a lot out there that we don't know about. That's more or less the size of the problem. Am I correct?
No, in our herbarium alone, we have 7.8 million specimens.
No, but what about species?
Oh, and so there's 400,000 known plant species, and there's a predicted 80,000 that we don't know.
Okay, 500,000. That's the idea.
Yeah.
Thank you.
Perfect. Miroslav, you had a question. I see Sarah's chiming in there. Do you want to ask that?
Yeah, I have written a few of them actually. Well, not posted them yet. It's for Barbara. The solution to your problem could be a combination of computer vision and sequencing. So basically, with computer vision, you filter out those that that computer vision is good with. So you just take out common species and then you basically sequence the rest. Because as you said, you have a few million specimens, and it's already feasible to sequence all of them. Not?
So when I was talking about the sequencing, it's sort of the things we can do with herbarium specimens that we couldn't do before. We couldn't have foreseen that we could actually, sequence genomes from a dried bit of plant. But as far as identifying species, we do, this you know, there's always, you know, you talk to any biologist, what's a species, you know, it becomes a very interesting conversation. But we do use, for some species, we have to use morphology and DNA sequence information. And this is, you know, also in sort of my dream world, the ability to link, you know, sequence data with specimen data would be amazing.
So I think my sort of follow-up question was a little more hypothetical, but it's more just like trying to think about, again, sort of like with these pipelines, like what are the effects? So if you're using a computer vision model to sort of take out all the common species first and then sequencing what's left, assuming that you've caught everything, I'm kind of just like, well, but like what if your computer vision model says that this is a common species, but in fact it's like a similar but very rare species? Like, I don't know, just thinking about sort of how that stuff might propagate.
I have a question for Olga. You know, with your background on, you know, crowdsourcing and data set collection and how influential that's been on the field as a whole, you know, have you recently sort of been thinking about kind of what is the next big step that the field needs to take, like in terms of the data and the problems that we should be looking at? And, you know, maybe to make it a little bit more relevant to this particular workshop, does fine-grained have a role to play in that? You know, is it as simple as like ImageNet again, but with much more fine-grained categories? Instead of, you know, 60 birds, we throw in 6,000 birds. Like what, you know, in your opinion, what's next for us? Like where should we be looking to go?
I think the next is moving more and more towards applications, towards sort of tying the tools that we're building closer and closer to applications. I think honestly there have been, you know, is it just going to be ImageNet for X? I mean, I think there have been many ImageNet for X's, but I think that's not where the big breakthrough is anymore. I think I think the next step is really moving towards what are the kinds of things in the world that we can tackle with computer vision? What are the kind of applications we want to build? There's a lot of visual data out there. There's, you know, very relevant to what's going on right now in the world. There's data in police body cameras and very little analysis being done of that data. There's data being, you know, more and more sort of hospitals, for example, are willing to put cameras in, you know, maybe it's not RGB cameras because you want to preserve privacy. Maybe it's RGBD or really just depth cameras. More and more work done on analysis of those, basically in sort of many important societal applications and problems. There's an opportunity for computer vision to come in. Honestly, you know, you asked about fine-grained. I mean, I think a lot of these applications do involve some kind of fine-grained analysis, right? If you think back to, you know, to body-worn cameras, for example, I think the analysis of the kinds of situations that get recorded there sometimes require very fine-grained understanding of, you know, human behavior and human interaction and looking for microaggressions and micro things that sort of lead to escalations. If you think about, you know, the hospital settings, there will be lots of, you know, again, very small differences that make a big difference. And so I think, you know, coming back to what I said earlier, that the fine-grained community has actually been, I think, ahead of, in some ways, the rest of the recognition field in, first of all, working with domain experts, and second of all, actually pushing visual recognition to really focus on these detailed differences between things which actually make all the difference in a lot of end applications. And so I think that's where we're going. I think that's why fairness is starting to become more and more important. That's why I think data set, I mean, there's definitely opportunities for crowdsourcing still and for, you know, large scale data, if nothing else, because what we know how to build now is models that work really well with large scale data. So there's of course opportunities for that, but that's, I don't think that's where the next big breakthrough will be, is who can collect the biggest data set. I think it's like identifying particular domains where we, with the computer vision expertise, could make the biggest impact.
Cool, thank you.
We have a few questions from the chat window to catch up on. So Pietro had one for Barbara about iNaturalist. Pietro, do you want to ask that one?
Right. So I was wondering, so we invested, I mean, it's here you have grant, and so we invested quite a bit of time collaborating with the California Academy of Science with doing iNaturalist. And now we're taking a big breath and saying, okay, was that the right thing to do and was it useful? So do you, when you look at iNaturalist, do you see something that could evolve into the tool of your dreams? Or do you feel like it's, you know, nice try, but it's completely in the wrong direction and needs something different?
I think iNaturalist is awesome, and it only works as good as how well the organisms were curated, how well were they identified in the first place, and that's what we kind of come back to with the herbarium. I mean, it's, you know, I mean, just to sort of even just take it even further out, the fact that we're still identifying new species of plants just never ceases to amaze me. I mean, we're talking about new species of palm trees. They're trees, they're not little tiny plants. So you know, iNaturalist and the herbarium models are only as good as the data that was used to train those models. Was it well curated? Were those identified correctly? And then you can get a good model out. Where we are with the herbarium, where I would like these models to go is, yes, to help curate specimens that exist that are known specimens, but also to find the new species more quickly, because that's the hard part. I mean, we have this room with 30,000, you know, unmounted, non-digitized specimens. It takes years to get through those, and by the time we get through them, it could be that habitat is, you know, completely burned down or underwater or something.
I'd love to follow up with a related question. Miroslav had a couple comments that he made, and that he works with insects. And, you know, I think one thing, just you mentioned, Barbara, 400,000 species of plants. And, you know, as you go to insects, you may have just the beetles alone, there may be 400,000. And then there's all these other families of insects and so forth. I think the scale is very different from many of the domains that we've tackled so far. When we're talking about, you know, we have a data set of 200 birds. It's kind of the de facto. Same thing with cars and airplanes and other things. You have these data sets in the size of hundreds. We're talking here about hundreds of thousands or millions of species. Just wonder if you have any thoughts. And it was quite interesting. Miroslav talked about. Do you want to just comment yourself, Miroslav, on this SMTP?
Yeah, I'm kind of good fit to this community because I have both backgrounds in computer vision and in taxonomy. So my supervisor is a biologist. He does computational biology, models of evolutions, and he ran a bunch of projects he collected insects, flying insects with nets, and he ran them for a couple of years. So he ran one large project for a couple of years and collected in a single project 50 million specimens. That's estimated. So it could be a few millions up and down. So they managed to get through 500,000 specimens in 15 years, and that's a huge success. He just published a paper in Nature. And he's just one of many successful scientists in biology who collect like large number of specimens. They can't sequence it because sequencing 50 million is just like it's a lot. If you go with, you know, just making a soup and then sequencing it as a soup, so you reduce the cost, then you can't go back and see what was in the soup. So you have to go one by one specimen, and while we are already handling them, maybe you should also photograph them and then make a model that filters out those that you probably could already identify with computer vision models. And when you watch it, like to one of these samples, it's like in 1 week, in one bottle, you collect 5,000 specimens. Usually like 5-6 species is already half of it. So it would be super good for people working with those, with insects, for instance, just to, you know, be able to classify and separate a couple of, but also to depict a couple of common species, because already that reduces their work in half.
So at Caltech, there's a professor named Joseph Parker, and he works with beetles. And one of the things that was sort of eye-opening for me was hearing how many of the beetle species like cannot be by a human classified without actually like dissecting their genitalia. And so like, how do you think that would play a role? Like, are there some of these where it's like there's a really common species, and then there's one that's not common, but they look visually identical even to a human? Or is that like, is it like a whole class of them that you just would know not to try to visually disambiguate?
There are several of such cases. Biologists will probably say there are many, and there are probably thousands, and there are many more because we are not aware of them. There was a study on the butterflies in Costa Rica where they thought it's a single species, but then they sequenced it and they saw it's actually at least 10 species. And then they figured out that each of these species actually also feed on different plants, but they never thought about it before, and they're completely separated genetically. They're more separated than like a duck and a chicken genetically. But just they look completely like same. Our vision is biased because we see things from one angle. Maybe, you know, nature developed different eyes and probably looks at things differently. You know? It's not everything in morphology. There's a lot of things in behavior, and like probably Barbara can say a few more words about these things. So there are many.
Yeah, I mean, you know, for plants, you know, distinguishing some species, you're looking at hairs, which are really small. For some ferns, you're looking at spores, like the structure on the spore, and a spore can be, you know, 10 microns across. It's like the width of a human hair. So there are some features that we need to distinguish some species. But I think Ryan really hit it on the head, is that, you know, with plants, with insects, with, you know, other organisms, it's a matter of scale. I mean, it's just that there's just so much we don't know, which is still really amazes me. But in order to, you know, if you're, you know, an expert in a plant family, you sort of have this encyclopedic knowledge about what you've seen already. So when you're going to another herbarium and you're going through sheets to see what's been collected that might be in your family, you sort of know what's been collected. But, you know, one person can only do a small fraction. This is where I think computer vision would be great for getting the scale down to something that is tractable for humans, to be like, okay, this is the pile to look at.
I would like to raise my hand, but I don't know how to do it here on this one. Just to blow your mind a bit to another fact. A few years back, there was a huge project in Germany where they also collected, but on many more locations, collected specimens of flying insects, so much bigger than what we had here in Sweden. So they ran it for a couple of years. They collected literally a lot. They could not even estimate how much they collected. So the only thing that they could do eventually was to sequence basically a few buckets and then to weight the rest. So they weighted the rest over the couple of years, and they basically estimated that they lost 50% of insect biomass. And that's huge, not more than 50, I think it's 70% of biomass, which is a lot. So we have no idea what we lost. We have no idea we know that we will lose, we're going to lose a lot in the future, but it would be also good that we at least know, you know, what we are losing, and then you know, to understand better these patterns, what's happening around us. And it's just a matter of processing more and more, faster and faster. Even things that we already are aware of, that we understand, like for a biological perspective, they're interested mainly in the long tail, you know, like those few things that are not seen before, or maybe seen only a couple of times, but not at that location, and so on. But in many cases, we also want to have a lot of what is already known, just to have enough data to understand patterns. Like for instance, we now know that migratory birds change their behavior they are coming earlier here in Sweden because we have data that goes back to 60s on yearly basis. We also know that same happens to butterflies. But we don't know for the rest of the insects. Probably there are some patterns. We also know that ladybugs, because they're seasonal here, they're actually hatching much earlier now than just 15 years ago. And this is just due to climate change. And we have no idea about the rest 1.999 millions of insect species.
Yeah, I sorry, just one last thing. Miroslav brings up a really, really important point is that, and this sort of goes back to what Pietro says, you know, do we need to do all the species? And I would argue that biodiversity is really important, understanding all the organisms within a habitat. So when you're seeing changes in insect diversity or butterfly diversity or bird diversity, you're not just linking it one, you know, maybe group of organisms at a time, but you're understanding that habitat. What other species occur there? Maybe the butterflies are coming earlier because the plants are flowering earlier, but we have those records in herbaria that tell us those plants were flowering in, you know, mid-June, and now we know they're flowering in early June, and we can track these different patterns. But to be able to see and to link that information across species would be amazing.
Okay, so I have a question to both for Barbara and Miroslav. So I'm wondering if so computer vision people are a bit lazy and we rely on images that are captured through cell phones or the internet and whatnot. And we don't go often out to capture images. And so one exception is, of course, Sarah with her camera traps. So one question is, would it be useful to have sensors in the environment that can measure those things that you're interested in, maybe not perfectly? And so I'm thinking, for plants, a pollinometer, which is, if you think of a camera with some vent that blows pollen grains against the surface and you image them. And for insects, something equivalent that, you know, if for insects, you know, they go through some tunnel or they crawl into some little chamber and they get imaged day after day after day. Should we invest time and effort in doing those things, or does it sound not so useful?
Shall I go first? Things for spores systems already exist. So this is something that has been counted manually until last couple of years, until CNNs took off. So there are already in place systems that basically count pollen spores, because some of those pollen spores might cause allergies. So that even before that, there were certain locations and institutions in like all over the world where they actually counted those spores. They classified them and counted them. So there was interest before for that, doing that, and I think at least in developed countries, systems are in place for doing that automatically. Probably not at the best possible performance. It could be improved. And for insects, there is no such thing. I'm writing a proposal for exactly that, and it should also include some of the acoustics.
So I've seen actually a few different like versions of like insect, like edge-based insect categorization. Obviously not for all insects, but when I was working at Microsoft, the health team there actually for epidemiology purposes built this crazy insect trap that has all these little cells, and what they're trying to do is capture specific species of mosquitoes. So they have little cameras in every cell that are cameras and also like vibration sensors that are categorizing the species of bug to make sure that they're only sort of shutting the trap when it has the sort of epidemiologically relevant species of mosquitoes, which I thought was incredibly complicated, but I guess is like a really good way for them at least to try to track the spread of disease. And then the other version I saw, there's a nonprofit in Germany called APIC, like I can type it in the chat, apic.ai. And they've built like a bee track that's categorizing bees, tracking their pose, trying to actually identify when they are sort of facing certain types of potentially like poisons from things like pesticides. And so trying to use bees as like a signal species for like the environmental health. So it seems like maybe it might be possible to bootstrap at least some of like the hardware and stuff off of these existing solutions and then just sort of change what the actual computer vision component is.
Yeah, I'm aware of these for bees. Several similar tools exist. I'm aware of those, but as I said, I'm not aware of any, tunnel where you can filter out everything that comes out and let's say separate those that are new, unknown, or your model is not confident about. No, such thing does not exist.
No, and that sounds super cool. I just knew that there was, I guess, some hardware that I'd seen.
Great. So I wanted to just real quick, we're a little over the time we anticipated and don't want to take advantage too much of our speakers. But if they're willing to stay around and others have questions, we'd love to, if people want to continue to chat, we can, but I want to take a minute and we can kind of virtually clap, but let's thank our speakers and panelists and for everyone for their participation and both great questions and thoughts shared as well. I think it's been a really great session. And so if anybody needs to take off, you're welcome to, but if you want to stick around, we can continue to discuss a little longer.
Thanks so much for having us. Thanks for running the workshop as well. Like, big thank you to the organizers.
Yeah.
Yeah. Thank you to the organizers and thank you to all the participants who participated in the Herbarium 2020 Challenge.
So I'll just echo that. And I want to thank all of the challenge organizers and all of the workshop organizers, all of our speakers from earlier in the day as well. And if you didn't get a chance to go watch the videos of their talks, please do that. They were great. So thanks everybody. We'll hang around anybody who can and wants to, but thanks again for joining us.